Okay, so last time we did digestive system. We talked a little bit about homeostasis and regulation in the beginning. We did nervous system. I left you guys an Easter egg on the action potential, so maybe watch that if you have time. Bonus questions on the exam could come from there. And then we talked about endocrine with hormones. And I gave you that list of hormones that you should be familiar with, a handful of them that you may see on the exam. So that leaves us with a few more systems to get through. So we'll start with musculoskeletal. The musculoskeletal system is one of my favorites. I don't know, this stuff is also interesting, but a lot of the muscle stuff we've already talked about, so we'll kind of get through this pretty quickly. But I want you guys to know, within the animal group, you've got 3 different types of skeletal systems. So you might see a hydrostatic skeleton. Can you guys guess from looking at the word hydrostatic what that might mean? What is hydro mean? Water. Yeah, do you guys know what static means, or stasis? I'll give you a hint. It's the opposite of dynamic. The dynamic and static are opposites. I know those are. So dynamic means changing and moving. So what do you think static means? Not moving, yeah, being still. So hydrostatic really means that the water holds you in place. Okay, so a hydrostatic skeleton, something like a sea star. Remember we said they have endoskeletal elements, but they don't really have a skeleton. So this is an example of a hydrostatic skeleton. That just means that their body cavity, their coelom, is fluid filled. And so that fluid is exerting pressure on the tissues and holding the shape of the organism. The muscles contract around the coelom to change the shape of that body cavity, and that's how these things produce movement. So they don't actually have skeletal elements that they are pulling on with their muscular system. So that's one way to do it. And then we've talked about exoskeleton and endoskeleton. Which one do we have? Endoskeleton. Yeah, that means your bones are where? Inside. Yep, your muscles are attached to the outside, wrapped around. Exoskeleton, the opposite is true. Muscles are attached inside and pulling in a different way. Something like an arthropod, like our ghost crab here, or Halloween crab, sorry. Ghost crabs are white. My bad. This guy's a Halloween crab. You can guess why, right? Orange and black. Pretty cute. Anyway, so those are just the 3 types of musculoskeletal systems. Muscular system and skeletal system are 2 separate body systems, really, but they work together so closely that we often sort of classify them this way. So what I want you to know about the skeletal system, we'll go through this really quickly. This is the only slide on bones that we're going to talk about, which is kind of a shame because bones are super interesting. But what I want you guys to know is that your skeletal system is made up of bones and cartilage. Okay, so both qualify as part of your skeleton. Functions. This is kind of interesting. So some of these you know. Some of them you may not be as familiar with, okay? Functions of your skeleton. So structural support and movement. That's a duh, right? You guys know that's what your skeleton does. It holds you up. It's the hard part to give you form. Without your bones, you'd just be a pile of soft tissue, yes? What about movement? Does your skeleton move by itself? It doesn't, right? So what's doing the movement? Which tissue type is associated with movement? Yeah, your muscles are doing the movement, but they're attached to the skeleton. So without your bones, you still wouldn't be able to move, even though your bones aren't moving. But if your muscles have nothing to pull on to produce movement, you're not going anywhere, right? So that's why movement is included in the function. Protection of internal organs. Like what? Give me some examples of skeletal elements that protect internal organs. Your ribs. Ribs, yeah. So, sorry? Brain, yeah, for sure, exactly. So hard protected cases over those soft organs. What about production of blood cells? Did you guys know that? Your blood cells, your red blood cells, white blood cells, and platelets are all produced in your bone marrow. So it's the site of blood cell production. And also regulation of calcium levels in the blood. So your bones store about 97 to 98% of all the calcium ions in your whole body. So when you need more calcium in your bloodstream to help regulate processes like heart rate and muscle contraction and things like that, you get it from that repository of your bones. So your bones release calcium. When your blood calcium levels are too high, they stop releasing calcium and store it up for you. So functions of skeletal system is important. There's also classification of skeletal system, which is worth taking a look at here. So you have an axial skeleton, which is shown in blue here on this guy, and your appendicular skeleton, which is colored red on the same image. So it's kind of interesting just to look at the two divisions because of what we talked about with paired appendages, right? So clearly we're looking at a human skeleton here. It's very self-centric in this particular part of the lecture. But you can see axial, what do you think axial means in terms of where you see these bones? What does the word axial indicate? Any guesses? Sorry? Axis, yeah. So the axis is the central sort of pole, right, that everything is revolving around. So that's axial. It's going to be skull, it's going to be vertebrae, it's ribs. It also includes ossicles of the middle ear. Those are the teeny tiny bones that are involved in hearing that you see in land vertebrates. Your hyoid bone, which is right up here in your neck. One of the only bones that doesn't articulate with other bones. Instead, it articulates at the base of your tongue. So it's holding your tongue in place in your throat. So your hyoid counts. And let's see, what else is important here? Skull, ossicles, hyoid, vertebral column, thoracic cage. So those all make sense, right? They're all attached. Now the hyoid and the ossicles, those are kind of weird. They're not really part of the axis as far as attachment goes, but they're considered axial because of location. Okay. Appendicular. What does appendicular mean, do you think? What is it associated with? Appendages, right? Those paired appendages. So you've got a pelvic girdle and you've got a pectoral girdle. That's your clavicles or your collarbones and your scapulae or your shoulder blades. That makes your pectoral girdle. And those girdles are attached to the axis, both pelvic and pectoral. And then your appendages are attached to those. Okay, so your appendicular skeleton is everything that isn't axial. So you've got bones of both of the girdles and then all of your appendage bones. All right, so just be comfortable enough to classify bones by where you would find them, either axial or appendicular. Make sense? All right, so functions and location. This we've seen before. This muscle slide is nearly identical to ones that you've seen before. I don't think there's any new information on this slide about muscle tissue. Are you guys okay if I just go on? Same stuff, same bold terms, voluntary versus involuntary, intercalated discs and cardiac muscle, all that stuff we've talked about. That's just review. And then I omitted the anatomy of the skeletal muscle, which also makes me very sad because it's really cool the way that your muscle cells contract to produce movement. But we'll talk about that another day or in another class. So the big red X means don't study that. Okay, you're not responsible for that information. That gets us to respiratory. So that's pretty quick, yes? You guys doing okay? Moving quick. I really want to make sure we have enough time to talk about immune, because that's what I wanted to sort of focus on this unit. All right, so respiratory system. What is the function of your respiratory system? What do you guys think? Well, breathing, yeah, moving air in and out. And then what's the point of moving air in and out? Oxygen, yep, gas exchange. So even if you're not breathing air, like aquatic organisms, they're moving water in and out, but they're doing the same thing. They're extracting gases from the surrounding media, and they're releasing waste into the surrounding media. So it's gas exchange. All aerobic organisms use oxygen to carry out their metabolic functions. Okay, so anything that requires oxygen. Respiration refers to how an organism obtains oxygen from the atmosphere. Typo there. So respiration is the gas exchange itself. Okay, the exchange of oxygen for carbon dioxide. Systems are adapted to their environment. That makes sense. You're going to look different if you live in the water versus living on land. Complexity of the system is correlated with the size of the organism. That also makes sense, right? So how does gas exchange happen? What chemical process do you guys think is at work here in moving oxygen in to the tissues or the blood and carbon dioxide out of the tissues or the blood? Diffusion, absolutely. And what did we say about diffusion over distance? Does it work well over long distances? It doesn't, right? So if you start getting larger and you start getting more complex, then you start to see the need for a more complex and intricate respiratory system in these organisms. So if you look at something like this aquatic flatworm, it doesn't have a respiratory system because it's flat. So diffusion works really well, just across its outer membrane, in and out of the water. So no respiratory system to speak of. But when you get up into something like an arthropod, you start to see some examples of these more complex respiratory systems. And arthropods like the bee, they have what's called a tracheal system. So you can sort of see it visualized here in blue. And they have spiracles, and spiracles are like little holes on the outside of their body that they can open and close to take in air and let air back out again. So they're essentially moving air in and out of this system of tracheae, plural, to get gas to different portions of their body, okay? And it's associated with this red piece here, which we're gonna look at in the next group, which is your cardiovascular system. And so cardiovascular and respiratory are really tightly connected to one another because both are involved in that gas exchange process, right? So air comes in from the air, or let's say oxygen comes in from the air or the water, and it's got to get to your tissues. So you're bringing it into your respiratory system, and then that exchange is happening between that media, right, either air or water, and the blood, and then the blood carries it out to the rest of the body. Okay, so you've got a really tight connection between respiratory and circulatory. But what I really care about the most when we're looking at these comparative type of groups, body system discussion things we're doing here, is thinking about these concepts that go back to that physiology chapter. They go back to thinking about form and function and adaptation to environment, and always with the surface area to volume ratio, right? So those themes you see played out over and over again. And then you get into something even more complex, like a fish, and you've got gill structures, which are even more specialized for gas exchange, also intricately connected with blood vessels, right, which are part of your circulatory system, cardiovascular circulatory system. All right, so that's respiratory. Let's talk about hours for just a second, because this is going to tie us really nicely into circulatory, which is coming next. So these two terms mean different things. Ventilation versus respiration. Okay, ventilation is moving the air in and out of the lungs. You can do that artificially, right? If you put somebody on a ventilator, it's just that machine that's pumping air in and pumping air out. For us, it's just a matter of changing the volume of your thoracic cavity. You guys know what your thoracic cavity is? What is that? What am I talking about? What's another term? What's another name for thoracic? Chest. Yeah, so just your chest cavity inside of your ribs. So this is your diaphragm. That's that large flat muscle that separates your chest cavity from your abdominal cavity. And when it contracts, it gets flat. So instead of being sort of arched like this, it comes down. When that diaphragm comes down, it opens the space inside of your chest cavity. And because the pressure inside of a container as it gets bigger goes down, when the pressure inside of your lungs drops, or I should say inside of your thoracic cavity drops, becomes lower than atmospheric pressure, air is going to rush down its pressure gradient into your thoracic cavity. So it's really, ventilation is really, you're just changing the size of this container, sucking air in, pushing air back out. Sucking air in, pushing air back out. That's ventilation. Respiration is where you see the gas exchange. Okay, that is exchange of oxygen and carbon dioxide between the air in the sacs of the lungs and the blood itself. That's external respiration. Then you get into internal respiration, where you get oxygen from blood to tissues. But that's a whole different subject. Okay, so all you guys have to know is the difference between ventilation, which is just moving air, and respiration, which is gas exchange. Okay, so you may see a question that asks you about that. So when you inhale, air is going to go through your nasal cavity or through your mouth, because they both connect to your trachea, which is this tube that leads down to your paired lungs. There's just a little bit of anatomy here. The trachea is a soft tissue tube, and it's lined with pseudostratified epithelial tissue that is producing mucus to keep foreign debris from being sucked all the way down into your lungs. So it's sticky, but the only reason it stays open for you all the time so that you can move air are these C-shaped rings. Ever wondered why your trachea looks like that? You can feel it in your own throat. It's kind of buggy. What you're feeling is these C-shaped cartilage rings that are holding that soft tissue open so it doesn't collapse on itself. Just because of surface tension, right? It's wet in there, it's got mucus, it's going to stick to itself. But those hyaline cartilage rings hold it open, which is kind of cool. So your trachea is going to branch into bronchi, which are going to go left and right and left bronchi, one to each lung. They're going to continue branching like the branches of a tree into bronchioles until they get smaller and smaller and smaller. And they end at the alveoli. And the alveoli are these single cell thick air sacs. They are, here's one alveolus, and they mostly are in clusters like this. So you get these alveolar sacs with individual alveoli in them. Each alveolus is wrapped in capillaries. You guys know what capillaries are? Tiny little blood vessels, also one cell thick. So you're talking about really, really thin tissue walls in your alveolus, okay? And really, really thin tissue walls in these capillaries. You've got air that's coming in through your respiratory tract, all the way down into these alveoli. So you've got oxygen-rich air inside, and you've got oxygen-poor blood outside in those capillaries. So after your blood is returning to your heart to get out to your lungs to pick up new oxygen, it's low in oxygen. It's coming back from the tissue, right? You've used it, so you've got deoxygenated blood or low sort of oxygen deficient blood that's wrapped in these capillaries that are wrapped around your alveolus. So you have a concentration gradient. You guys see this? High oxygen inside the alveolus, low oxygen in the blood tissue, in the capillaries, in the blood. All you have in between that blood and that air is a layer of tissue, one cell thick in the alveolus and one cell thick in the capillary. What does that make easy? Diffusion. Yep. So all that's happening is simple diffusion, you guys. It's an incredibly elegant system. Works really, really well, very simply. So you've got a high concentration of oxygen. It's going to diffuse into the blood and get carried away back to your heart to go back out to the rest of your body, carry oxygen. And carbon dioxide is the opposite situation. It's high in the blood, right? Because it's coming back from the tissues. So it's picked up all that waste from your cellular respiration in your tissues. And it's low inside the alveolus because you're breathing in fresh air, yes? So your concentration of carbon dioxide is reversed. It's going to move down its concentration gradient just by simple diffusion out of the blood into the air. And when you exhale, you push it out of your alveolus, through your bronchioles, through your bronchi, out the trachea, and out the mouth. Pretty elegant, right? Pretty cool system. All right, so here's a little box that explains everything that I just said in a lot fewer words. So that's the summary of what's going on. Air containing oxygen crossing lung tissue, bloodstream, blah, 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 blah. Whole summary, okay? We're gonna look at the cardiovascular system or the circulatory system here in just a second and sort of tie the two together. But before we do that, I just wanna show you guys this. This is one of my favorite infographics I've ever found on the internet. If you sit here and stare at this long enough, you'll become an expert in respiration. Now, it's just a really cool example of the differences in the way that different animals respire. So you've got our lungs up here. It is just tracing the path of oxygen and carbon dioxide. So the yellow is oxygenated and the carbon dioxide is red. Did I say that right? Fresh air yellow, old air red is what I'm trying to say. So you can sort of follow and see what's going on. And here it's labeled for you, oxygen yellow, carbon dioxide. So you can sort of see what's going on. Our system is got basically you inhale and you exhale and you're not ever doing two things at the same time. But you've got other animals that can do it a little bit more efficiently. Birds are incredibly efficient at moving air because their lung doesn't actually inflate and deflate, but they have air sacs, accessory air sacs that are outside of their lung. So they inhale and they store fresh air in those air sacs that gets pushed through their lung as they're exhaling carbon dioxide. So the two never cross paths. The two types of air, oxygenated versus sort of deoxygenated air, if you will. Oxygen and carbon dioxide don't mix together. So you're always pumping fresh air. Watch, oxygen in, oxygen across. Oxygen in, oxygen across. So even when you're breathing carbon dioxide out, they're getting oxygen in. Why do you think that's helpful for birds? Why do you think that's an adaptation that you see in bird respiratory systems? Yeah, they're high metabolism, right? Super high metabolic rate, super high energy demand, requires a lot of oxygen. So they're really good at breathing for that reason. And then this is showing a grasshopper. So this guy has a tracheal system, and it's kind of showing you how that works. Okay, so you can just sit and stare at this. Maybe when you're having trouble sleeping, you can just like watch it. It'll hypnotize you and fall asleep. All right, so there's no new information on this slide. It's just cool. Okay. All right, so that brings us to circulatory system. Functions to transport nutrients and waste. Okay, so your circulatory system is carrying those gases, oxygen and carbon dioxide, but it's also carrying nutrients like vitamins and minerals and macromolecules that you've broken down from the food, and really anything that you could possibly need. It's also carrying waste. So not just carbon dioxide, but things like nitrogenous waste and other products that you're making through metabolic processes. The complexity of your circulatory system correlates with size and complexity of organism. We just said that about the respiratory system, right? It's the same reason. So when I say no respiratory system equals no circulatory system and ask you why that would be, you should be able to come up with an answer, right? I'm going to put you on the spot and make you tell me in a paragraph what the answer to that question is, but you get it, right? If you aren't complex enough to require a respiratory system and you're operating under simple diffusion for gas exchange, do you need a circulatory system of vessels and a pump to carry stuff around your tissues, do you think? You don't, right? It's the same reason, right? So this complexity of circulatory structures kind of goes hand in hand with the complexity of respiratory structures. Make sense? Okay. All right. In organisms that have gills or lungs, which are more complex respiratory structures, a circulatory system is necessary to carry oxygen to tissue. You've got farther to go. You've got more tissue to supply. You've got to have a way to get it there. And just like with respiratory, diverse forms of circulatory systems have evolved to meet the needs of different body types and sizes. I don't really care too much about showing you this piece, but this is what I kind of wanted to show you guys, because we just looked at the tracheal system. We'll go back to that here. So you can sort of imagine that blue tracheal system overlaid on that dorsal blood vessel here. So this guy has multiple hearts. There are little pumps along the way, and it's pumping hemolymph, because this is an open circulatory system, so it's not closed like ours. All of our blood is contained inside of vessels and pumped around your body by a heart, right? This guy, his circulatory system is open because it's not a closed circuit, okay? And so it's got multiple pumps, and it has what's called hemolymph instead of blood, because it all mixes together in arthropods. Still more complex than like a flatworm who doesn't have one at all, right? But not quite as complicated as when we talk about our human circulatory system. So we'll talk about this, then we'll do some comparison, and that'll be it for circulatory. It's a similar pattern as you saw with respiratory. All right, you guys ready to take a journey through your circulatory system? Let me go forward one and just show you. Here's us, okay? This is mammalian circulation. Does this look more complex than, say, a fish or a salamander or a snake? The evolution of complexity, right? Here we are. So this is the most complex system of all of them. So we'll start with the hardest one, and then we'll talk about the differences, okay, in our comparison. So you have a heart and you have blood vessels. That is your circulatory system. It's moving blood around to feed your tissues. Remember, it's carrying nutrients, and it's also functioning in gas exchange. You have a 4-chambered heart. Okay, when we get to the other organisms that we're going to look at, you'll see the differences there. And 4 is the maximum number of chambers that you see in any animal heart. So we have a 4-chambered heart, 2 atria, 2 ventricles. Okay, and they're all doing sort of different jobs for different purposes. Pumping blood through 2 separate circuits, and everything is separated. Okay, so you don't have any mixing of oxygenated blood with deoxygenated blood, which is a really efficient way of doing things. We're going to talk about why that works well when we talk about these comparisons on the next slide. So you have your systemic circuit and your pulmonary circuit, and they're separate. So your systemic circuit refers to your entire body system, blood circulating through your body, all your body tissues, carrying oxygen and nutrients to your tissues and cells, and waste products, including carbon dioxide, away from tissues and cells. Okay, so we're talking about your systemic circuit, you're talking about all of your tissues. Okay, when you talk about the pulmonary circuit, you guys know what pulmonary means? Lungs. This is the circuit of blood vessels that takes blood to the lungs to get oxygen and back to the heart to enter the systemic circuit. Okay, so blood has to go through both circuits, and there are valves in between each of the chambers. So in between atria and ventricles, and in between ventricles and blood vessels, and all the way through you have valves to keep blood flowing in one direction so you don't get backflow. Okay, so it's super efficient. We'll look at these two circuits here in just a second, but first I want to just kind of touch on this last little statement here. Large arteries and veins branch into smaller and smaller blood vessels until they reach capillaries. We've already been talking about those capillaries, tiny little blood vessels with walls that are 1 cell thick, which makes diffusion possible. So gas exchange with the tissues happens at these beds of capillaries. So you have so many capillaries strewn throughout your tissues that so they're right. The flow into capillaries is regulated. So they're not always full of blood. They only open when your tissues actually need fresh blood. But if all of your capillaries opened at once, your blood pressure would drop and you'd die because there's not enough blood in your entire body to fill up all your capillaries and your other vessels at once. That's how many capillaries you have. It's pretty astounding, actually. So what's going on with your circulatory system? We'll just take blood through circulation really quickly through these two circuits. So let's start with deoxygenated blood coming back from the body. Okay, so all of your body tissues are making waste, generating carbon dioxide, and they're picking up oxygen. So when blood returns from systemic circulation, what do you think the concentration of oxygen looks like when it's returning from systemic circulation? High or low? Oxygen. Low, right? What about carbon dioxide? High, right. So it's coded blue. Okay, the color code is blue here. So we're looking at systemic, this is systemic circuit. Now it's a cartoon drawing. So imagine that these vessels and these capillary beds are representative of all of the other millions of capillary beds that you actually have in your systemic circuit. So this piece is representing your lower body and all of your systemic circulation, and this one is representing all of your upper body circulation. Okay, so just imagine that these extend further than what they do in this cartoon. But what's happening is you have blood that is returning to circulation from the lower half of your body to this vessel, your inferior vena cava, drops down into the right atrium, the first chamber, okay, that we're going to talk about. You also have a superior vena cava. It's also connected to the right atrium. It's draining the top of your body, your head and your arms, and it's landing here in the right atrium. So you've got deoxygenated blood landing in the right atrium. Atrium contracts, pumps blood into the right ventricle. Okay, so that's 2 chambers of the 4 so far. Right ventricle is going to contract and push out into pulmonary circulation. Okay, so these are your pulmonary arteries. These are where deoxygenated blood is heading out to the lungs. And what's happening at the level of the lungs? We just talked about this with the respiratory system. Why is it blue up here and red down here? What's going on? Yeah, it enters with high carbon dioxide and low oxygen, picks up oxygen at the lung, right, with exchange with the air, that's respiration. And then this blood that's leaving the lungs is now full of oxygen, right? And it has dropped off carbon dioxide. So your concentration has switched because of this diffusion down across opposing concentration gradients, okay? So that's all happening in the lungs. Now from both sides, remember you have right and left lungs. So from both sides, this blood is coming back to your heart in through your pulmonary veins into left atrium. Okay, so that's chamber number 4. That's the receiving chamber basically from pulmonary circuit, because now you've got oxygenated blood in your heart Left atrium contracts, blood goes into the left ventricle. Left ventricle contracts and pushes blood out into your aorta, which then branches into all the rest of your systemic arteries, taking that healthy, oxygenated, low carbon dioxide blood out back into systemic circulation. That's a lot, right? What I want you guys to do, to be able to do, is just tell me what's going on in both of those circuits. Okay, I'm not necessarily going to expect you to trace the flow of blood through the heart, okay? But I just wanted you to see what's going on with these 4 chambers and these 2 separate systems, because it's complex. But once again, an elegant dance, right, of blood moving through these 2 systems. Does that make sense? So you have 2 completely separate sets of circulatory vessels, one for pulmonary circulation, one for systemic circulation. That's pretty complicated when you start looking at comparing across groups. So when you look at something like a fish, now how does a fish breathe? What kind of respiratory organ does a fish have? Most fish. Gills, yep. So if you look at the structure of their circulatory system here, they have a 2-chambered heart. That's much simpler than a 4-chambered heart, right? And blood is only moving through in one direction. But that's because when blood passes through the gills, they're connected directly to their systemic circulation. The gill arteries go directly into systemic. Okay, so the only thing the heart is doing here is pumping in one direction, out to the gills, out to the gills, out to the gills. Make sense? So deoxygenated blood in fish is going out to the gills, getting oxygenated, and then going straight from gill arteries into circulation. It's a little bit simpler, but it works well because it's an aquatic organism. And water is filtering across the gills just by they open their mouth, right, and swim. Okay, through those gill slits, water flows out. Okay, so just flowing across those gills, gas exchange is still happening, similar to what happens in your lungs, but you don't need that 2-chambered heart because you don't have 2 systems. When you get into amphibians, it starts to get a little bit more complicated. So you've actually got 2 atria and 1 ventricle. But if you look at the colors that they're using to code oxygenated versus deoxygenated blood, what do you think purple means? If blue is low oxygen and red is high oxygen, what is purple? Yeah, both are there. It's mixing. Okay, so they've got 2 atria that are separate. So why do they have 2 atria? What do they have? What do these amphibians have that the fish don't have? Lungs. Yep. So you're starting to see that differentiation. Remember, fish crawled on the land. The first terrestrial vertebrates are amphibians, right? And then reptiles come later and mammals are the youngest, right? So you're looking at the evolution of complexity in the circulatory system and the respiratory system. So along with lungs comes that requirement for 2 atria, because you're pumping from heart to lungs and from heart to body. So you start to see that separation, but you don't have full separation in the ventricle, so they mix, okay? So in terms of efficiency, mixing oxygenated with deoxygenated lowers that efficiency of the system overall. Does that make sense? Because your good fresh blood is mixing with your old blood, so you're sort of like decreasing the oxygen by way of dilution. You guys with me? So it works for amphibians, okay? When you move into reptiles, you see a little bit more complexity, 2 atria and 1 ventricle, but that ventricle is partially divided. So you're starting to see the septum that's coming together to keep these things completely separate, which you ultimately end up seeing in the mammalian circulation system. Guess who else has a 4-chambered heart just like us? Birds. So yes, birds are reptiles, remember, living dinosaurs, but they are they have the same setup of circulatory system as we do. Why? Goes back to this same idea here. What do birds do? What do birds and mammals have in common that reptiles do not have in common with either? As far as energy and bioenergetics. Are birds endotherms or ectotherms? Endotherms, just like us. What about the rest of reptiles? Ectotherms. Yep. So it's energy requirements, right? If you are going to have the highest metabolism of any land vertebrate, you probably need a super efficient circulatory system, just like you need a super efficient respiratory system, because high energetic demands means you need a ton of oxygen because you're an aerobic organism and you use oxygen to do cellular respiration, which allows you to produce ATP, right? Everybody with me on this train? Okay, great. I don't know, I said musculoskeletal might be my favorite, but cardiovascular is pretty interesting too. I don't know, it's all pretty fascinating. You are a magnificent machine, aren't you? All right, excretory and osmotic regulation. This is not that interesting. It's kind of interesting, but you know, it's really mostly about waste. It's about making sure that you get enough of the water out or retain enough water, homeostatic balance of osmotic balance, and also that you have the right solutes, the right balance of things like salts and other minerals that are going to keep you alive. Okay, your excretory system includes structures from other body systems. So it includes your skin because sweat counts as an excretion of waste. It includes respiratory because CO2 is a waste. It also includes your nitrogenous waste from your urinary system. Okay, so excretory system includes all of these things. We focus in on the urinary system here because we haven't talked about that yet. It's pretty straightforward, you guys. Paired kidneys that filter your blood, make sure that you keep water when you need to keep water or get rid of it when you have too much. Hang on to those solutes that you need, get rid of those you don't, flush out things like, metabolites from medications that you take or other things that you ingest. Making sure that you keep amino acids, sugar, vitamins, blah, 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 when you need them and get rid of them when you don't. Basically, it's selective filtration that's going on. 2 kidneys, ureters that lead down to a storage unit called your bladder, and then 1 tube out for urine. Okay, that's it for circulatory. So I might ask you to recognize the structures. Did I just say circulatory? I meant excretory. I might ask you to recognize the structures and the function for this system, but that's pretty much it. Okay. Which gets us to the real exciting part that I want to talk about. Immune. So this is going to be laying the groundwork for our discussion on Wednesday. How much do you guys know about your immune system? A little bit? A ton? Any experts? Anybody interested in immunology? A little? All right, well, you should be, okay? At least enough to understand how your immune system works. The vertebrate immune system is unique. Like many systems, it is complicated, and there are parts and pieces in our vertebrate immune system that don't exist anywhere else, particularly in our adaptive immunity. So the part of your immune system that allows you to remember infections and fight them more efficiently later, that is uniquely vertebrate, okay? So that's what we're going to really spend time talking about here. We're going to look at innate versus adaptive immunity, and then we're going to look at the parts and pieces of each in a little bit more detail. So innate immunity, do you guys know what the word innate means? It's like an innate talent. It's just there. You're born with it, okay? So innate immunity is present and functional at your birth. You are already able to fight off pathogens, foreign invaders, if you will, at birth. Your innate immunity is rapid and non-specific, meaning anything gets in, it attacks. Okay? It doesn't have to recognize it. It doesn't have to remember it. It doesn't have to mount a response. It just goes. Okay? And it is also localized. So it's going to take action at the site of where that invader comes in. If it's a cut in your skin, or some sort of other type of tissue injury, or the initial place where viral particles get into your mucous membranes, that's where it's happening, local, okay, right at this site. And no memory. So it just does its thing. It does it well, but it doesn't remember, it just attacks constantly, okay? It's also constantly surveilling. So you've got cells that are just floating around looking for stuff, waiting for the opportunity to engulf something, or punch holes in something, or call in other cells to help, or whatever the case may be. So it's wandering around and keeping a guard. 3 categories under innate immunity include barrier defenses, chemical defenses, and cellular defenses. We'll talk about each of these a little bit. So your barrier defenses, that's your first line of defense. What is a barrier? What's a barrier? Give me an example of a barrier. Doesn't have to be biological. Like, what do you do around your yard if you want to keep other people's dogs out or your dog in? A fence, right? What if you live in a castle and you have to dig a trench around your castle? What is that? Moat, right? That would be a barrier. What about an umbrella? Is that a barrier? From what? Right. So barriers are just things that keep other things from getting through. Yes? So your barrier defenses are, first and foremost, your skin. Works really well. You got lots of layers. Remember we talked about how it's stratified squamous epithelial tissue? So you got lots of layers to sort of keep stuff out. It's also keratinized. So it's got this tough, waterproof protein in all of your skin cells, protecting you from the outside world. And then mucous membranes are basically skin that's on the inside, okay? Your mouth, your nose, the inside of your digestive tract, anywhere that the external environment can come into contact with your internal environment can be lined with mucous membranes, okay? And they have lots of cool things that they can do. Your skin does stuff like your sweat contains antimicrobial compounds. You're basically sweating hand sanitizer, low levels all the time. Your saliva has enzymes in it, antimicrobial enzymes. Your tears, antimicrobial enzymes. Okay, so you're like a walking factory of sanitizer, okay, coming out of these defenses. Mucus itself is a defense. How is it a defense? What does it do? Mucus do. Let's think about mucus in your respiratory tract. Is it sticky, do you think? What's it catching if it's sticky? Bacteria, viral particles, dust, pollen, hair, debris of any kind, right? It's all catching in your respiratory mucus so that it doesn't get all the way down into your lungs, right? So loogies and boogers, you know, you guys know what I'm talking about. Those are all just collections of stuff that gets trapped in your mucus and then you expel it to get rid of it so it doesn't go in, right? But if you do happen to swallow some of it, your stomach acid will take care of it there, right? So you've got a lot of things going on here with these sort of barrier defenses. Chemical defenses include your inflammatory processes. So you have all kinds of chemicals that are released from cells that tell other cells to freak out. They call in other blood cells that come in to fight things. They raise the temperature of your body to make your own body less hospitable to pathogens. So inflammation is going to include things like redness, heat, swelling, and pain. Okay, all of those are caused by these chemical defenses of your innate system that rush right to the source, that localized place, right? So think about like a cut, okay? It doesn't just bleed, but you also get swelling and pain and heat because of all the things that are rushing to that site. But fever is also part of inflammation. It's an inflammatory process in and of itself. It's caused by inflammatory chemical defenses called pyrogens. Okay, so fever is going to raise your body temperature. What I want to talk about here is the idea of adaptation. Okay, so any organism is going to have pathogens that are well adapted to living in its body, right? So organisms that are well adapted to humans have evolved alongside humans, and they really like 98.6 degrees Fahrenheit, right? What is 98.6 degrees Fahrenheit? Yeah, about the normal body temperature for a human being. So what do you think happens if you raise that temperature by a few degrees? What if you get to 102? What do you think that does to the pathogen that's well adapted to maximum efficiency of enzymatic function at 98.6? What happens when you raise the temperature of the environment to a chemical reaction? What happens to the chemical reaction? It can or slow down, right? So it's messing with the optimal operating temperature of enzymatic reactions in that pathogen. Ultimately, it's screwing with its environment, right? So if you can heat it up enough to where that pathogen is compromised in some way, you can slow its ability to infect your tissues, and that's what fever is for. Now, if it gets too high, that's problematic for you, right? If you start getting too high, you're going to start denaturing your own enzymes, okay, and your own proteins and causing yourself problems. But that's part of your chemical defenses. There are cells as well that are involved in your innate defenses. They're non-specific. They're just wandering around. Macrophages are the wanderers that eat stuff. They're big and they go around engulfing things. And what they do two different things. So they might engulf something and then just fuse it to a lysosome. And inside of a lysosome, you've got lysozymes, which are digestive enzymes. So you basically just eat something and then digest it. You've taken care of the problem. Doesn't matter what it was. You didn't have to call in any reinforcements. You just ate it. Okay, that's what some macrophages do. Other macrophages operate as what are called professional antigen presenting cells. So they'll eat something, then they'll break it apart and they'll take pieces of it and they'll stick it on the surface and like wave it around like, hey, look what I found. This is bad. Hey, come help. And it's calling in other cells of your adaptive immune system to come in and do things. Fight it, kill it, make antibodies, whatever the case may be, okay? So you've got macrophages that are doing that, and you've also got natural killer cells, which are some of the coolest. I mean, if you were a cell, wouldn't you want to be called a natural killer cell? I'm pretty good. Hardcore, right? Anyway, what these guys do is wander around, and if you have body cells that are acting weird, they take care of the problem. So if you're virally infected and you don't look normal, natural killer cells go to work. Cancerous cells, natural killer cells can detect those as well. So anytime a cell is sort of acting weird, natural killer cells will do one of two things. Both of those pathways are going to end with apoptosis. Do you guys remember what apoptosis is? Cell death. Yes, programmed cell death. So natural killer cells can wander up upon a virally infected cell or a cancer cell, notice that something weird is going on, and hit them with a compound that triggers apoptosis from the outside. Or, this is my favorite part, they can pour enzymes called perforins on the surface of the cell, which essentially punches holes in the cell membrane. And then once there are holes in that cell membrane, then they inject enzymes called granzymes, which trigger apoptosis from the inside out. So ultimately, they're just going around killing stuff because it looks suspicious. That's what it does all the time. Starting at birth. Pretty good system, right? But this is not unique to vertebrates. The part that is unique to vertebrates is the adaptive immune system, okay? And that's going to include your B cells and your T cells. You guys heard of B cells and T cells before? Probably have, just in sort of passing. Adaptive immunity is developed as a result of exposure to antigens. So antigens, it's an interesting word. So pretty much any cell is going to have proteins on its surface that are considered antigens. They're just recognition proteins. They're markers. They're identity proteins. But what your immune system can do is recognize those identity proteins on things that aren't supposed to be there. And when it's not supposed to be there, it mounts a response. So antigens trigger an immune response if those cells are not supposed to be there. Your own cells have antigens on the surface, but your immune cells are trained to recognize that that is you. So those are what we call self-antigens. So all cells have antigens, but your immune system is supposed to only recognize those that are foreign. All right, and that's the job of what your adaptive immune cells are going to be doing. Adaptive immunity is specific, meaning that it will only attack if it encounters the right kind of antigen. So your B cells and your T cells are genetically programmed to recognize one kind of antigen. How many kinds of antigens do you think exist in the world? Just a wild guess. Like 10 or like 10 billion? What do you think? Trillion, right? Any number of things. If every cell is displaying something that could be considered an antigen, you've got trillions of varieties. But every one of your B cells and T cells is genetically programmed to only fit like a lock and key with one particular type of antigen. That's pretty astounding too. So you have the genetics to make immune cells that recognize anything, just hanging out in there. So you have like one or two of each, okay? For a whole bunch of different antigens. The only ones that are going to react are those that actually encountered their specific antigen. It's kind of a freaking miracle that it ever happens at all, right? But what happens is a T cell let's say T cell, for example, comes along and it finds that antigen that it is genetically programmed to respond to. And that T cell is like, oh, it's my moment to shine. And that T cell is stimulated to divide and multiply. So it makes a clone army. It's called clonal selection and clonal expansion. So one cell is selected because it fits that antigen. Let's say it's the spike protein on the surface of COVID-19, right? SARS-CoV-2. So you have a cell, or a couple of them, that are circulating around through your system. Maybe it's in your lymph node, maybe it's in your bloodstream, who knows? It's just circulating and surveilling, and it comes across that spike protein and it fits like a lock and key, and that particular cell is activated and it goes, and it proliferates and makes a whole bunch of different cells that are designed specifically to fight that antigen. Nothing else, just that. So specific, and that's what adaptive immunity being specific means. It's only going to attack one thing. That attack is systemic. So when those cells proliferate, divide, and go on to do their jobs, they're doing it in your entire system, not just at the site of the infection, because things can spread, right? Pathogens spread. You have the ability to develop memory. So these cells, both T cells and B cells, after they've differentiated, some of their downstream progeny, some of their cellular products are memory cells. And those cells hang out even after the infection is resolved, so that if you encounter that same antigen again, next time, it's fast and it's stronger. So fast and so strong that you may not even know you encountered it. That's how vaccination works. You guys with me? Okay, we're going to talk more about that in just a second. As I mentioned, your adaptive immunity exhibits tolerance to self. It's called immunocompetence and immunotolerance. So to be competent, you have to be able to attack things that aren't you, but to be tolerant, you have to be taught to not attack things that are you. And it's a very complicated process that your T cells go through in your thymus and your B cells go through in your bone marrow that teach them the difference between self and non-self. So that's one sort of early step in the process. Once they're taught to recognize self versus non-self, then they go out into the lymphatic system and hang out, wait to encounter an antigen. You've got B cells and you've got T cells. T cells are going to do a lot of different things when they are selected and they expand into this clonal army. Some of them are going to become helper Ts, and helper Ts essentially secrete chemicals that call other cells in, or activate B cells, or make other cells do things. Okay, they secrete cytokines, which are like activation chemicals for other cells. Cytotoxic Ts. What do you think cytotoxic means? Can you guys tell by looking at the word? What does cyto mean? So they just kill cells. They're kind of like natural killers. They act the same way. They use the perforins to punch holes. They use the granzymes to induce apoptosis. They work very similarly, but they're a different type of cell. Suppressor Ts are there to turn it off. When the whole thing's over, when the fight is done, the suppressor Ts say, okay, everybody, mission accomplished, knock it off. So you don't have an overactive immune cell, and then you've got memory Ts. So remember what happened, so next time they can do it fast. B cells, these are the real rock stars. Well, they're all kind of rock stars, but these are the guys that are going to make your antibodies. And antibodies are the chemicals that are going to neutralize the pathogen. They're going to tag them for destruction. They're ultimately going to keep that pathogen from being able to do any harm. Okay, so when you activate this whole adaptive system, sorry?
Because my sister had mine.
I don't think they're talking to me. Muted. Okay. Yeah, so when this whole system is activated, some of your B cells are going to also be clonally selected, clonally expand. Many of them are going to turn into plasma cells. Those plasma cells will start cranking out antibodies, okay, to start binding those antigens, but you're also making memory B cells at the same time. So you have T cells, memory T cells that remember what your Ts did the first time, and you have memory B cells that remember how to make antibodies. So the second time you see that same pathogen, you can do it, you can make those antibodies a lot faster. All right, so let's look at immunological memory, because that's going to get us to our vaccine discussion for next time. So B cells are activated by cytokines to become either plasma cells or memory B cells. That's what we just talked about. Your plasma cells, again, are the ones that are making those antibodies. And the memory cells are going to hang around, pretty small numbers of them, in case you ever encounter anything again. The first time you encounter that pathogen or that antigen that's going to interact with those immune cells, it's your primary immune response. Okay, primary first, right? When that happens, it takes a long time to get up to your titer, your antibody titer, which is like the concentration of antibodies in your bloodstream. So it doesn't get as high, right, as it will on a secondary immune response. So the first time you see it, you get a little bit of an immune response. It's enough to start making antibodies. It's enough to differentiate those B cells into plasma cells and memory cells, but you might get sick. Okay, so let's say you get exposed to SARS-CoV-2. It's novel to your immune system. You've never seen it before. That is your primary immune response. It might take 2 weeks for your B cells to get up and fully functioning and pumping out antibodies enough to fight off that infection. So in that 2 weeks of time, you feel pretty crappy, right? You know that something's going on. The virus is making you sick. Your immune response is making you feel sick, right? Because remember, some of those symptoms that you're feeling, like inflammation, right? Fever, achiness, that type of stuff, that's all from your innate system. So that's just you fighting. That's just you feeling bad because you're fighting. So that happens the first time. But the second time you encounter that, you are not likely to get sick again because your memory B cells are there and they can start cranking out antibodies right away. So your second exposure, boom, antibody titer way up, way past where it got the first time. And not only does it get higher, but it does it faster. So you may never even know you were exposed. That's the idea, right? So that would be naturally acquired active immunity, where you make antibodies because you got sick one time and your body remembers what happened. But the idea behind vaccination is artificially acquired active immunity, meaning you are exposed to something that can't make you sick, but is enough to trigger your immune system to start making antibodies against that antigen. So that if you do ever encounter it in a natural infectious type of situation, it's not enough to make you sick, it's just enough to trigger your secondary immune response. So you start cranking out antibodies right away, you may never even know you were exposed. Does that make sense? All right, here's what antibodies do. This will be the last thing we'll talk about. So we'll effectively eliminate the reproductive system from the conversation, which is kind of my plan anyway, unless we had extra time, which we will not. So antibodies, when they're produced, they interfere with pathogens, as I mentioned, by neutralization, which means they bind to those cell membrane binding sites. In the case of coronavirus, it's that spike protein that we keep hearing so much about. Antibodies will bind to that and prevent it from entering the cell. Okay, so that's how the spike protein is the protein that the virus uses to get you remember, it's been a while. Remember when we talked about viruses and we talked about how they use your own cell membrane receptors to get in, so they lock into there. In the case of SARS-CoV-2, it's your ACE2 receptors. They're all over the place and lots of different cell types, which makes this virus so particularly generalized. It can get into lots of different cell types. But your antibodies, these little Y-shaped tubes, will come in and bind to that particular site on the virus, so it can't get in to your cell. Okay, so that's how antibodies work through neutralization. It can also use opsonization, which means they coat the pathogen and tag it for phagocytosis. So here's your big wandering macrophage that eats stuff. Now it's been coated with antibodies that opsonizes the pathogen. It says, eat me, essentially, and the macrophage comes and does that. Okay, so that's another way that antibodies can work. Antibodies can also work by activating complement, which is part of your innate system that we didn't even talk about. But basically, complement ramps up that same type of action. Marks pathogens for phagocytosis, makes it easier for them to be eaten. Some complement will actually cause lysis of cells. They can form something called the membrane attack complex. That's almost as hardcore as natural killer cell, right? Membrane attack complex. Anyway, so complement works in hand with antibodies. What I want you guys to know from this whole slide is basically that antibodies can work in different ways, but the one we're talking about, the antibodies that you will produce if you are vaccinated against SARS-CoV-2 are neutralizing antibodies. So that's how those work. So they basically go in and they bind to that spike protein, and then viral particles can't get into your cells. And if they can't get into your cells, they can't do what? The virus. Reproduce, replicate. Right? Because viruses are obligate intracellular parasites. Remember, they have to get into your cell and use your genetic machinery, your ribosomes, to start making viral proteins and assembling viral particles, virions. So they can't get in, they can't do that. And if they can't do that, they can't make you sick. And if they can't do that, they also can't spread. Right? So the idea is to get your body to make neutralizing antibodies without making you sick. And we do that through vaccination, right? So the two videos, so here are the three videos that are here. I want you guys to watch these before our conversation on Wednesday. They're less than 10 minutes each. You guys can skip reproductive altogether. This stuff's kind of interesting, but I'd rather talk about immune. So don't even worry about these slides. I'll go in and put a red X on those and repost the PowerPoint, but don't worry about reproductive. So I want you to watch those three videos. Then I want you to watch the other two videos that are posted on D2L. We'll look at those again real quick before we disperse today. So just in case you weren't here before, or just to review, on the homepage, go to class discussions, mRNA vaccines and immunity. All 5 videos, including the 3 crash course videos that are linked in the PowerPoint, are also linked here. These 2 videos, mRNA vaccines explained, this is less than 7 minutes long. It's going to explain the technology behind the vaccine. The first 2, the Pfizer and the Moderna, that are both the first 2 that got emergency authorization for use. They're both mRNA vaccines. So this is going to require you to call back what you have learned today and what you'll review in these crash course videos on immunity. Also call back what you know about messenger RNA, transcription, translation, particularly translation, because you get to skip the transcription part because you're already at messenger RNA. So just watch this. It's pretty straightforward. It's not like a science video, so it's made to be understandable. And then this one is about vaccine side effects. So you can think about why you would get those side effects and if that's a bad thing or a good thing. Okay, so I'm looking forward to having a discussion about putting all the pieces together. So what you're looking for is basically like, what is this vaccine doing? What is it making? And what it's making, what is that product doing to your immune system? Okay, so those are the kind of things that we're going to talk about on Wednesday when we reconvene. Okay, it's going to be helpful. This is my plan, my big devious plan, is to make it easier for you to answer questions. Because you guys know as well as I do that there are millions of questions and lots of doubters out there now that say, I'm not getting it, I'm scared, I hear this, I hear that. And your job as a scientist is to dispel the darkness, right? To bring light to the situation. Yes, to bring information. So you're well equipped to answer questions or to address misconceptions. You choose to. Now you don't have to engage with every person on the street who's hollering about being an anti-vaxxer or whatever. But if you have a conversation and you wish that you knew more, you'll know more after Wednesday. That's the goal. Okay. All right. That's what I got for today. And that wraps us up for exam material, which won't open until Wednesday. So you got some time to study and look back over videos and whatever you need to do between then and now. And it won't close till Thursday night at midnight. So you've got some time on that too. All right. Thank you for your attention, as always.